And off we go. Plant of the day is something called dodder, not daughter, not like, you know, daughter of a person, but it's dodder, Dodder. Do y'all see the yellow or orangish kind of stringy stuff back here?
We cannot see your screen.
Cannot. Can nobody see my screen?
I see it.
There it goes. Okay.
I will try to reshare. Okay, can everybody see the screen now?
Yes.
Awesome. Okay. So we see all the orange plant in the background. That is a vine that is non-photosynthetic. It actually is a little, I would call it a vampire plant, the way that it sucks the life out of other plants. I've only seen it once that I can remember. It's not really all that uncommon. I just haven't been in the right places, I guess. But I remember one time in graduate school, the places where I lived in the university were on either side of a river, and you would just cross the bridge on the sidewalk. I remember looking down in the river one day because it wasn't really high then, and thinking, who threw out all this like orange weed eater line? I really thought that someone, I didn't know that it was an actual plant. When I saw it, I really thought it was something man-made, just kind of some kind of string thrown out. And it's a very interesting plant, in my opinion. Not the best picture here on the left, but I love the one here on the right. It is a flowering plant. It has flowers. So the top two pictures are before and after. This is where the daughter is actually connected, and it really likes tomatoes. There's some other plants too that it likes, but you can see where it actually inserted itself into the phloem of the plant. And so in that phloem, it's able to connect and then take away nutrients that plant is doing through photosynthesis. It's got a lot of different common names. The scientific name is Cuscuta, which is the genus, and there are multiple species. But I love these non-scientific names. Devil's guts, devil's hair, devil's ringlet, gold thread, hellweed, hairweed, hellbine, love vine, pull down, strange weed, witch's hair, witch's shoelaces. I get the impression people don't like this plant very much based on the name. And it can be very destructive on crops if it makes it into your field. And on some occasions, seeds make it into grains, or seeds that are going to be used in growing like some kind of crop. And as your crop grows up, so will this, and it basically is it's hard to deal with, and a lot of times you have to scrap your crop because it's too intertwined, literally. Here's a tree that's covered in it, and that's just fascinating to me. It kind of looks like a fuzzy tree, but it's just covered in dodder. Okay, let's ratchet things up a notch. We've got to talk about some of the tissues, and this should start to sound repetitive because you've heard some of these terms before. We'll take them into more depth. Meristems are permanent regions of active cell division in plants. We don't have meristems in animals. We may have areas of active cell division, but not in the same format that plants do. So these are areas where cells retain the ability to divide throughout life. A lot of our cells in animals, we don't have active cell division. There are some organs that do and some organs that don't. Plants have some that do and some that don't as well. The ones that do are called the meristems, and there are different meristems, one of which, probably the most important or prevalent, are going to be what you're calling the apical meristems. Apical, think about the term apex. These are found at the tips of roots and shoots, and that's where active growth and tip of roots and shoots are. Why is this important? Well, if you cut off the apical meristem, you're going to stop growth at that part of the plant. Okay. Now there may be another apical meristem that can start to grow from there on out. We'll We'll talk a little bit about how those can get activated, but there are some plants, for example, like palm trees. Why do we have a problem growing palm trees here in, Columbus? People do, but there's not a lot. Why do you think that is? Okay, it gets too cold, Elena says. Why do you think cold is a problem? Like what does the cold do to cause an issue for the palm tree? Elena's heard this one from me before, right? um, so the answer is actually that it does have one meristem. So in the situation of palms, they don't have any protection from the cold, or very little protection from the cold to protect their one meristem. They have a single meristem at the top in which they continue to grow up. Unlike most of the other plants around us that have multiple meristems. And if some of them were to get frozen, some of the ones that are more protected can activate and continue growth later. But palm trees don't have that. They have one. And most of the time, at some point they'll get too frozen to be able to survive here for very long. Although I do know a few that grow around here. Now we also have sago palms, which have something similar as a problem. They're not actual palms. And then we have saw palmettos, which are palm related, but they are adapted to living here and are just fine. But it is all about the meristem. You freeze the meristem and you kill it, you don't have growth from that meristem at least. So meristems increase the length at the apical meristems and produce new cells. There are 3 primary meristems. These are things you want to know for sure. There is the protoderm, there is the ground meristem, and there is the procambium. We're going to go into more depth with these, but there are 3 primary meristems. There will be secondary meristems that will occur, but these are the primary ones. We want to make sure that we understand what that means. Protoderm develops from your main apical meristem into your dermal tissues. For example, the epidermis. Okay? And hopefully that name makes a lot of sense. Apical protoderm, apical protoderm. So derm, epidermis. If you're familiar with stem cells, for example, in animals, there are 2 kinds of stem cells. There is embryonic stem cells, which can develop into any tissue in a human body or other animal body. And then there are adult stem cells. Have y'all heard those terms before? Embryonic versus adult stem cells? I can't tell if anybody. Okay. So adult stem cells are different than embryonic stem cells because they have a limited range of things that they can become. So you get adult stem cells from certain tissues in the body, and they can become a number of cells, but not everything. And that's kind of what I see apical meristems and the primary meristems being. Apical are like your embryonic stem cells. They have something called pluripotency. And primary meristems, I think you can use the term totipotency. One is you can become anything. One, you have a limited range of things that you can become, but there's still a lot of cells that you can become. So pluripotency versus totipotency, apical meristems versus primary meristems. Now the ground meristem does never become tissue that's dermal. It becomes what's called ground tissues, your parenchyma, collenchyma, sclerenchyma. It's called the ground tissue system. We've already heard about these in lab. And then you have the procambium, and these are the ones that produce what's called primary xylem and primary phloem. There will be secondary phloem and secondary xylem to come. So the procambium or the cambium system will become your vascular tissue. So protoderm is the epidermis. Ground meristem are your ground tissues. Procambium are your vascular tissues. We want to know that. So when you think about a meristematic cell, it really can become anything. It can become a vessel element. It can become a fiber, okay? And these are sclerenchyma. They can become parenchyma. They can become sieve tube. Actually, I'm sorry, not the vessel. Some of these will be involved in your xylem, but they are sclerenchyma tissues that are dead at maturity, which we're going to get into. Now, outside of your primary meristems, you have things called lateral meristems that can create secondary growth. So these are tissues that increase the girth. Does anybody know what I mean by girth of roots and stems? If I increase your girth, what would happen to you? Okay, your thickness, the way, yeah, you get bigger around. That's girth. So we know that trees' trunks get bigger around. Okay, same thing happens in roots as well. So trunks are stems, roots also underground will do that. One of the secondary growth systems that happen in lateral meristems happens at the vascular cambium. It is something that is considered to be what's called bifacial, and bi meaning two, facial meaning kind of face or side. There's two sides that it grows from. It actually grows on both sides of the cambium, and it produces secondary tissues that function primarily in support and conduction. Secondary phloem is produced to the outside, that outside face, and secondary that's backwards. I realize I put this in here backwards. Let's fix this, okay? Make a note. Secondary phloem is actually produced to the inside. No, I'm sorry, I do have it right. My goodness, got it backwards in my head now. Ugh. Monday morning. That's all I can say to that. No, it's right. My goodness, why am I doing that? Ugh, my goodness. Okay. Secondary phloem is to the outside. Secondary xylem is the inside. Shooey. Secondary xylem produces your wood, and you get pieces of those secondary xylem every year left behind. You get more and more and more. This is what we see with the rings on the inside of your tree trunk. And have y'all seen tree rings before? So Kyrie's seen them, okay. Everybody else is good with it too, right? Okay. Thank y'all for the heads up on that. That's what the secondary xylem produces. And it does this every year. And every year you get a new ring. At least in the deciduous forests that we live in, the temperate region. Secondary phloem, on the other hand, is produced to the outside, and we get new phloem every year because the old phloem gets destroyed every year. And most of these things are very thin cylindrical cells because they act as pipes, if you want to think of it that way. It's pipes for flowing things up and down. Sorry guys for getting that backwards in my head. Can't believe I did that. Cork cambium is another lateral meristem, and it's outside of your vascular cambium. So if you're thinking about your tree, okay, and it's going to get bigger, it's going to get bigger because of the vascular cambium, but just on the outside of that is another set of growth areas, another meristem that actually produces more bark because the epidermis cannot be replaced. Epidermis is primary and it's not replaced in a secondary fashion. So what do? We have to protect the plant on the outside. So we produce something called bark, which is produced from the cork cambium. So it replaces that epidermis for those plants that have have secondary growth. Basically, if you think about, there's a certain amount of surface area that epidermis has, and when you stretch it, it's going to break eventually. And when that happens, we start replacing it, not with more epidermis, but with the bark that we see on trees. Here's a couple of examples of where you can see your meristems in the shoot. So here's a shoot apical meristem, and here's a root apical meristem. I like to call them Sam and Ram. Sam is your shoot apical meristem, S-A-M. Ram is your root apical meristem, R-A-M. And they are protected by various things. Down the bottom, it's a root cap. On the top, it's actually new leaf primordia. And you see them kind of overlapping in protection. So this little bit of space right here is your apical meristem. That's where all the growth takes place. Everything other than that, it's not growing anymore. It's just the cells getting larger. There's a difference between getting larger and growing. Growing has the connotations of cell division. Getting larger is the cell physically getting larger, stretching, elongating. Does that make sense for everybody? The difference between growth and getting larger. Okay, because we'll see that especially in the root, because you'll see cell division down here, which we've already seen some of in our onion root tip. As you move further up into the elongation region in the maturation area, you actually see the cells get bigger, longer in a lot of cases, and they become mature, especially their secondary cell wall becomes more thick. Here's an example of what the cork looks like, the bark. So your dead epidermis is stretched and broken on the outside. You have a layer of cork cambium, phyloderm, and then right inside you eventually hit the cortex. From phyloderm to the cork, this is basically your bark, okay? And the cork out here, not really alive. It's very waterproof, as we'll see, to protect that plant. You also have intercalary meristems, and you don't see this in every plant. When you don't have a vascular cambium or a cork cambium, and that makes a lot of sense in these grasses, you don't produce bark in grasses. Even though sometimes we think of bamboo as kind of tree-like, it's not. It doesn't have bark. In fact, it doesn't produce wood either, so that's the vascular cambium. But they do sometimes have a secondary one called the intercalary, and near your nodes, these areas where the leaves are attached and the growth is taking place, you have an extension, like basically it grows the internode longer. And that's what you see here. The internode's actually increasing in size because the intercalary is growing it longer. This happens in grass. It's one of the reasons why we have to mow the grass. So let's go into our simple tissues. I would suggest, just like you made a table for the algae, you might want to end up making a table for your tissues. And you'll need to think about your organization if you do that. Simple tissues. These are tissues made up of only one cell type. This is the parenchyma. That's the first one. So we're looking at the ground tissue. This is composed of parenchyma cells. They have very large vacuoles, and sometimes they contain different secretions, especially in those vacuoles. All plants have parenchyma, all. They are the least specialized, very most basic cell type. I wrote call there, my goodness, my typing is awful. They are both primary and secondary in origin. Well, what on earth does that mean? Well, it means that they can be produced from a primary meristem, and they can also be produced from a secondary meristem. Okay, something like your vascular cambium could produce some parenchyma. They have a primary cell wall only, whereas we'll see differences in other ones. They're also very relatively thin, and we've seen this in lab. They're large cells. They are living at maturity. They have a very thin cell wall. When you're looking at them through the microscope, it's very bright because the light can make it all the way through with very little obstruction. And some of their purposes are to be involved in food and water storage, and some of them are more involved in photosynthesis. So some can be pith, and some can also be involved as like mesophyll cells that do photosynthesis in the leaf. So you can see how open and bright it looks when you look here. For example, there are some specialized forms of these too that are not shown in this picture. But there are something called aerenchyma, and this is parenchyma tissue that has connected air spaces, basically to make them floatable, like you might see in the nymphaea. That's a water lily. The chlorenchyma is parenchyma cells containing chloroplasts. So we see them as like what we might call a mesophyll cell later that does photosynthesis. So very simple tissues. Second is the collenchyma, and they contain living cytoplasm, so they're also alive and they can live for quite an extended time. They're variously distributed among plant groups, so not as ubiquitous as parenchyma. They are also primary and secondary in origin. They have a primary cell wall only, but in this case, it's not a thin cell wall. It's relatively and unevenly thickened, thicker than what you see in parenchyma. And sometimes there are places where it's thickened more than in others. And you can kind of see this under the microscope. They are living at maturity, and their purpose is for mechanical support and flexibility for organs and food and water storage. So there's some crossover in their jobs with parenchyma. Now, can you see that this one looks a little less bright? And it's because you get some thickening of the cell wall, and it's going to block a little bit more of the light when you're looking through. So it's going to look a little darker, maybe a little bit less resolution because of the reduction of light under the microscope. Here's some of those intercellular spaces that we might see that make them more buoyant in some cases, especially when you have large ones. These are just spaces that are in between. So there are kind of a mixture here of collenchyma and parenchyma as we move kind of inwards. The larger cells are probably, let's say those are probably parenchyma. It's hard to tell here, but usually when you have this layer of collenchyma, it looks smaller and more compact than what you see with your larger parenchyma. But you're going to see there's a layer of this, like when you move from pith into the cortex, you'll see a kind of a boundary area like this. Sclerenchyma, these are sometimes called fibers. These have cells that are very thick, very tough, secondary cell walls. You don't always have them. They're not always present. They can also be secondary and primary in origin, and they have a secondary cell wall. So they have the primary cell wall like the other two and a secondary cell wall. That's pretty thick and is usually what's considered to be lignified. It's what we think of as becoming wood. They are dead at maturity, a very different consideration when you think about collenchyma and parenchyma because they're living at maturity. These are dead. So they mature, then they die, and then they're used. And they are for mechanical support, and a lot of times associated with parts of the plant that need to have more strength, especially around with our more complex tissues, as we're going to get into, like with your phloem and your xylem. Here is a special kind. I wanted to show you this with the hair, and I didn't get a chance to do this. We may still try to bring it in someday because I would like to show it to you. But these kind of crystalline kind of looking structures, These are specialized sclerenchyma that are called sclerids. And if you've ever eaten, have y'all all eaten a pear at some point? What would you say is one of the big differences between pear and apple versus not including taste? What about the texture?
It's grainy.
It is. It's grainy, and that is the what do you call it? The sclerids that you're tasting or feeling, I should say. Also, when you think about like peach pits, the hard pit that's around, that's also developed from sclerids, just a concentrated amount of sclerids. Okay, let me see. Here are some fibers, and you can see how thick. Now, these are cross sections. So for example, like if you're thinking this is your fiber going up and down, when you cut it this way to look at it from above, that's what you're seeing on the left. On the right is from the side if you're looking at it. But when you do a cross section, you can see just how thick things they can really get on these secondarily thickened cell walls. Okay. Let's see. Okay, epidermis is another simple tissue. So we've got, this is a dermal tissue this time. Outermost layer of cells that cover all exposed surfaces for new plants. They're found in all plants. They are primary only, primary only. And that's really going to mean that they're produced early on, sometimes versus later on, which is a lot of times your secondary meristems. So let me clarify that. Primary, I say early, but I'm really referring to things that come from your apical meristems. If it develops on the plant later, away from the meristems that are apical and shoot and root, then you're going to be dealing with the secondary meristems, a lot of times those lateral meristems. Okay, so it's primary in origin only, primary cell wall only, very relatively thin. They are living when functional at maturity. They are usually, and I say usually, here it says one cell thick. We are going to see examples where they have more than one cell layer thick. So the epidermis can have just one cell layer all the way around, but we have a few that live in dry habitats that have more than one. Most of them, but not all, will produce cutin to form the cuticle, that waxy substance. And if you've ever had a houseplant, you can sometimes tell the leaves are kind of waxy feeling, especially like they get dusty and you're going to wipe them off or something. But you can tell they're very waxy. That's the cuticle that makes it waterproof on the top and to some degree on the bottom. On the roots, some epidermis cells will become root hairs, and we see that over on the top left. On the leaves, some will become stomata. They'll actually become your guard cells, and they will be the only cells that are epidermal in origin that have chloroplasts because they are, sensitive to light, whereas your regular epidermal cells out here don't have chloroplasts. You can also produce something called trichomes, and we might call them hairs on many of the plants. Like if you've ever touched a fuzzy plant and you're like, oh, that's hairy. They don't have hair. They're not mammals. What they have are trichomes, and they are modified epidermal cells, some of which can be kind of spiky. You can see a picture on the bottom right here. By the way, real quick question. What kind of microscope took this picture on the right? Any ideas? Just to review your test one material. The scanning electron microscope, yeah, the SEM. On the left, these two are probably TEMs because you can actually see inside of the cell. So what you'll see for most of the epidermal cells are to deal with water loss or water absorption in the case of roots, and to protect against pathogens. Sometimes with these hairs, you're going to see that they're connected to glands, and on occasion the the hairs are kind of shiny, and when you're looking at the plant, and they may be glandular, and they're actually excreting little substances that shine in the light. So that may also be something that happens, but usually it's about protection. Hairs are about protection from losing water loss. So the epidermis produces a cuticle to protect against water loss. Hairs are produced to protect against sun and getting overheated, and also water loss, and of course also pathogens. I don't know about y'all. I don't really want to eat fuzzy lettuce. It's just not a texture I want in my mouth. The only corollary to that is going to be your roots, which are do the opposite. They're about a water absorption, but it's still all about water. So can we pretty confidently say that plants really, really care about water? Most definitely. Okay, you also have dermal tissues called the endodermis. And so the epi is on the outside and the endo is on the inside. And these are generally present only in the roots, and they surround your vascular tissue. And we'll use the word steel in the future, but the steel that runs down the middle of the root gets protected again from water loss. They're primary in origin. They only have a primary cell wall. They have radial walls that have a different kind of waterproofing substance. It's not cutin, it's called suberin. And suberin deposits will appear in strips, and they're called Casparian strips. I truthfully have a hard time seeing these under the microscope, to be truthful. We'll try to find them in lab, but just heads up, they may or not be so easy to see. They are living at maturity, and the whole purpose is to reduce lateral movement of water and nutrients, so going out and about. So here's a leaf. You can see the epidermis up here, epidermis here with a cuticle layer on top. A lot of times you can kind of tell the lower cuticle layer is thinner than the one on top. And it's because the sun comes from above. We worry about water loss more there than on the bottom. And then of course we do not put cuticle on the opening in our stomata down here. Okay, because we want gas exchange. If we seal it over with a waterproofing substance, we can't get gas exchange either. Okay, there are some air spaces, aerenchyma. Most of these cells inside are mesophyll, which are chlorhynchema. Here is a area that's a vascular bundle. These vascular bundles, we haven't gotten into those tissues yet, the xylem and phloem. But most of this stuff is parenchyma in some form, or epidermis. I usually see cortex, which is collenchyma in the stems. I don't notice it as much in the leaves, although you might get some associated, maybe some of these here that are more unevenly thickened, kind of around your veins of vascular tissue that kind of protect them. You might see it there, but probably not just out and about in your leaf. We've also got periderm as a dermal tissue, and this constitutes your outer bark. This is where you get cork cells. You also get suberin produced here as a waterproofing mechanism. And of course, how on earth do you get gas exchange through these thick bark waterproofed systems? Well, instead of having the epidermis with guard cells that open and close, they produce this area with parenchyma cells that basically are little projections through and into the periderm that are called lenticels that are used for gas exchange. Where's my picture? There's my picture. I want to show it to you real quick. So here's a lenticel. And so you'll get gas exchange. It's kind of a break in the bark in a lot of ways. And we're going to see these little things on the outside of the stems today in the lab. Let's go back. So here's that thick bark. We've already seen it, the cork. You can see the layers of kind of suberin in between that's waterproofing things. It's being produced from the cork cambium. And then dead epidermis is on the outside. Here you can actually see some of the differences. So the cork is on the very outside. The cork cambium is that lateral meristem that's producing cork and also producing phelloderm. And the cork cambium phelloderm is your periderm. Okay, I want y'all to know these terminologies. So if we talk about periderm, we're talking about all three of these things together. Okay. When we talk about bark, we're including those three things as the periderm and the secondary phloem. Okay. So if anybody actually pulls the full bark off of a tree, you're actually pulling off one of the vascular tissues. Now, if you just pull that outer layer off, a lot of times you're not pulling all the bark. You're just getting either the periderm or parts of the periderm, maybe the cork. You know, so hopefully if y'all have ever played with the bark on a tree, you haven't picked it too deep that you've gone into the phloem. If that happens, it can actually be detrimental to the plant, because just like if you get a cut in through your skin, you can get an infection. Same thing can happen with a tree. Do y'all have any questions? No, okay. I'm going to stop there. I want to do the secondary tissues later. I think that's a good spot. We're going to see some of this in the lab today as well, as we look at stems. We will talk about some of that secondary growth that we didn't get to today, but we'll get to it on Wednesday. So lab and lecture are almost lined up for the moment. It won't stay that way, but it works pretty well this week. Okay. Everything else okay? Okay, well, I'm going to let y'all go. Elena, stay on. And I don't think I have anything until I see y'all at 2. Just make sure you study for your quiz, okay? Be prepared. Okay, I will see y'all later, and I'm going to stop this recording.